So, imagine you take a completely naked bacterial cell, right, and you just drop it into a glass of fresh water.
Oh, it would be catastrophic for the
Right.
cell.
It basically instantly explodes. Like, the internal pressure is so massive that the cell just rips itself apart.
Yeah, it literally pops.
And so the only thing keeping the entire microbial world from just, popping into oblivion every time it rains is this single invisible microscopic suit of armor.
It's an incredible piece of biological engineering.
It really is. And that is exactly our mission for this deep dive. We are unpacking this high-pressure, microscopically violent world of the bacterial cell wall.
Specifically, this miraculous molecule called peptidoglycan.
Exactly. So, okay, let's unpack this. We have this huge stack of sources covering everything from the foundational chemistry of this armor to, like, some really gross 19th century accidents.
Always the best kind of scientific accidents, honestly.
Oh, totally. And we'll get into the evolutionary arms race happening around it right now. So if you're listening, setting the big picture here really changes everything about how you view modern medicine.
It absolutely does. I mean, understanding this microscopic wall isn't just biology trivia. It is the fundamental reason our most famous antibiotics actually function.
To why they fail. Right.
Exactly why they fail, and how clinical diagnostics still rely on these chemical parlor tricks from over a century ago. The stakes of this architecture are literally life and death.
Okay, so before we can break the wall or, stain it, we have to actually visualize how it's built.
Right, which is where a lot of people get tripped up.
Yeah, because the sources keep using the word wall, which obviously conjures up an image of stacked bricks.
Right.
But reading through the actual chemistry, that is like a terrible way to visualize it.
It's completely misleading. Peptidoglycan, or sometimes it's called mirin, is actually made of these alternating sugar units.
Right. You have N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM for short, thank goodness.
Yeah, NAG and NAM is much easier to say. So they link together in these long strings, and then you have these short peptide chains that stitch those sugar strings to each other.
So it creates this massive cross-linked structure, meaning instead of separate bricks just sitting on top of one another, you have a three-dimensional mesh.
Yes, a mesh is the perfect word for it.
I kept thinking of it like, well, not a brick wall, but more like a chain link fence.
Yeah.
But not even a flat one. It's like a single microscopic chainmail onesie that forms this perfect unbroken sphere wrapping the entire cell.
A chainmail onesie. I actually love that analogy.
Right, because it's not made of separate disconnected pieces. It is a single, continuous, covalently bonded bag-shaped molecule.
The sacculus.
Yes, the sacculus, which is just wild to think about. Like back in the 1950s, researchers took these bacterial cells and they hit them with heavy detergents to dissolve the membranes.
Where it wash away all the internal proteins and everything.
And they were left with these ghostly hollow bags, the sacculus just perfectly holding the original shape of the living bacterium.
It's amazing. And the physical reality of that chain link sphere gets even stranger when you look at the specific amino acids they use to build those crosslinks.
Oh, this is the Jack Strominger stuff from the mid-20th century, right?
Exactly. Strominger and his team mapped this out. What's fascinating here is that bacteria build this peptidoglycan mesh using D-amino acids.
Specifically D-alanine and D-glutamate.
Right. And for anyone listening who doesn't obsess over biochemistry,
which was probably most people,
the weird part is that they're using D-amino acids, which are the right-handed mirror images of normal molecules. The rest of the biological world runs almost entirely on the left-handed L form.
So all of our proteins, all the normal stuff is left-handed.
Exactly. So these bacteria are actively choosing the quote-unquote wrong building blocks.
Which begs the question, what is the mechanical advantage of doing that? Why be the odd one out?
Well, it essentially creates a molecular stealth mechanism. You see, biological molecules have chirality, that handedness we just talked about, and enzymes in nature work like highly specific locks and keys.
Okay, so the enzymes that predators or competitors pump out into the soil or like your gut, they're designed to cleave normal left-handed L-amino acid bonds.
Exactly. So when those environmental enzymes bump into this right-handed D-amino acid wall, they literally cannot find a grip. The physical geometry just doesn't align.
Oh, wow. It's like building a lock on your front door that requires a mirror image key.
That's a great way to put it. No predator is walking around with that mirror image key.
So by using these bizarre building blocks, the bacterial wall becomes effectively invisible to ordinary digestive weapons.
Completely immune to them.
Okay, so the architecture is super stubborn chemically, but we keep mentioning this massive internal pressure. If we jump into the actual physics of the cell, Why does a bacterium even need this rigid pressure vessel to survive?
It all comes down to osmosis. A bacterial cell's cytoplasm is just packed with solutes.
Sugars, proteins, ions, all that stuff.
Right. It is a highly, highly concentrated jelly. But the outside environment, like, say, a puddle of fresh water or even human blood, is far less concentrated.
And the rules of thermodynamics dictate that water desperately wants to cross that semi-permeable cell membrane to dilute the jelly inside.
Yes. And this inward rush of water creates immense internal turgor pressure. We are talking about pressures equivalent to or even exceeding a fully inflated car tire.
A car tire.
Yeah.
Inside a microscopic cell. That is, that's terrifying, pushing outward from the inside of it.
Constantly pushing. And without the sacculus, that chainmail onesie acting as a pressure vessel, the plasma membrane just yields to that pressure and tears apart. The cell lyses.
It pops. Which, honestly, this brings up a major shift in how I understood antibiotics after going through these sources. Because, like, penicillin, right? And that whole class of beta-lactam drugs.
The heavy hitters.
Yeah. Wait, so they don't actually poison bacteria the way, like, arsenic poisons an animal?
No, not at all.
They don't just shut down the cell's metabolism and kill it?
No, they actually turn the cell's own physics against it. Beta-lactam antibiotics just block the final stitching step of the wall construction.
The enzymes doing the stitching, the transpeptidases?
Right, also known as penicillin binding proteins, or PBPs. Penicillin chemically mimics the natural building blocks of the wall. It slides into the active site of that cross-linking enzyme and just permanently jams it.
So the bacterium is sitting there trying to grow and divide. It's synthesizing new membrane. It's taking in water. The car tire pressure is building.
But it can't finish weaving the pressure vessel around itself.
Oh, man. So the cell is essentially opening up holes in its own armor to expand, but the penicillin prevents it from patching those holes back up.
Exactly. The internal pressure rushes to the weakest point, the membrane bulges out through the gap in the chain link fence, and the cell pops like an overinflated balloon.
That is wild.
And this is precisely why penicillin is only effective against actively growing bacteria. If a bacterium is just dormant, not trying to remodel its wall, the pressure vessel remains intact.
And the drug literally does nothing.
Exactly.
The idea that we use their own physical pressure as a weapon is just fascinating. And, it really echoes one of the most famously gross historical moments in our sources.
Oh, the Alexander Fleming story.
Yes. So 1922, which is actually 6 years before he discovered penicillin, Fleming is in his lab, he has a cold, and he accidentally sneezes directly onto a bacterial culture plate.
Which is terrible lab hygiene, by the way.
Right, just awful.
Yeah.
A drop of his nasal mucus lands right on the bacteria, and instead of tossing the plate like a normal person, he just, watches it.
A true scientist.
Exactly. And the bacteria growing near the mucus literally just start melting away.
Because he had stumbled onto lysozyme, which is an enzyme that's super abundant in human tears, saliva, and, yes, mucus.
And lysozyme acts like molecular scissors, right?
It does. It specifically targets the glycosidic bond. That's the chemical link between the NAG and NM sugars that form the backbone of the peptidoglycan mesh.
So when the lysozyme cuts those sugar bonds, the chain link fence basically just falls apart.
Yep. And once again, without the structural integrity of the wall, the internal pressure takes over and bursts the cell.
Dissolve the mesh, and the pressure does the rest. But, okay, if Fleming saw the wall melting from mucus in 1922, how did we figure out that penicillin specifically causes this, construction jam, rather than just acting as a generic poison?
Well, that mechanical proof came from Joseph Park in the early 1950s. Park was studying staphylococcus cells that had been treated with penicillin.
Okay.
And he noticed that the dying cells were hoarding these massive amounts of a specific, really small molecule inside their cytoplasm. It actually became known as the Park nucleotide.
So they were basically stockpiling raw materials.
Yes, it was a massive molecular traffic jam. The Park nucleotide is a stalled precursor. It's a building block of the peptidoglycan wall attached to a carrier molecule, just waiting to be transported across the membrane and stitched in.
Ah, I see. Because penicillin jammed the final assembly enzyme on the outside, the entire production line backed up on the inside.
Precisely. Finding that stockpiled precursor was the smoking gun. It proved exactly how penicillin disrupted the mechanical construction of the wall.
Okay, so if penicillin's entire trick is popping this specific pressure vessel, that kind of assumes all bacteria wear the exact same armor.
Right.
But if that were true, a single antibiotic would just cure absolutely everything, right? There has to be a structural variation in how they build these walls.
Oh, there's a massive architectural divide. And to understand how we map it, we actually have to travel back a bit further to 1884.
The Berlin morgue.
Yes, Hans Christian Graham, a Danish physician, he's working in this morgue examining lung tissue from pneumonia victims, and he's really struggling to make the bacteria visible under a microscope against the backdrop of human cells.
So he just starts experimenting with dyes. He hits the tissue with crystal violet, then an iodine solution, and then he tries to wash the excess color out with alcohol.
And the results were completely split.
Right. Some bacteria trap the deep purple dye perfectly, but others lose the purple entirely. They wash clean, and he has to counterstain them with the second dye, which turns them pink.
And what's funny is he actually publishes this as an imperfect trick. He has no idea why it works. He just knows it helps him see the cells.
He apologized for it in the paper. And he accidentally invented the Gram stain, which is like the most critical diagnostic test in all of microbiology.
It really is. But it took another 70 years for science to catch up to the mechanics of his accident. Because the Gram stain is not a biological family tree, it is a real-time chemistry test of wall thickness and lipid content.
Okay, so let's break down the mechanics of that. If both sets of bacteria have this Peptidoglycan chain link fence, why doesn't the alcohol wash strip the purple dye out of both of them? Is it just that one is thicker?
Thickness is the primary factor for the purple ones, yeah, the gram positives. They have massively thick peptidoglycan walls, like anywhere from 20 to 80 nanometers.
Just dense layers and layers of that chain link fence.
Exactly. And this thick mesh is sitting entirely on the outside of their single plasma membrane. Plus, it's reinforced with these polymers called teichoic acids.
Which act like rebar in concrete.
Right.
Anchoring the layers together and giving that thick wall extra rigidity.
Right. So when Graham applied the iodine, it bound to the crystal violet dye, creating these large, bulky clumps inside the wall. Then, when the alcohol hits that incredibly thick teichoic acid-laced wall, it dehydrates the peptidoglycan.
So it shrinks.
Yes. The pores in the chain link fence shrink and close up, which permanently traps those large dye clumps inside.
It effectively locks the door on the purple dye. But the pink ones, the gram negatives, they can't lock the door.
No, because their architecture is completely different. They have a whisper-thin layer of peptidoglycan. Sometimes it's literally just a single sheet.
Wow.
And it's sandwiched in a space called the periplasm between two distinct membranes. They have an inner membrane and a really lipid-rich outer membrane.
Okay, so when the alcohol wash hits a gram-negative cell,
it rapidly dissolves that fatty outer membrane. And because the underlying chain link fence is so flimsy and thin, it just can't trap the dye complexes. The purple color washes straight out.
and then the pink counterstain is applied so you can actually see them, which logically, makes you think that gram-positive bacteria with this massively thick rebar-reinforced wall must be way more dangerous.
You'd think so.
Yeah, it seems like they would be so much harder for the human body to fight off than a bug with a flimsy single-sheet wall.
It sounds totally intuitive, but virulence, the actual danger a pathogen poses, does not correlate with wall thickness. In fact, gram-negative bacteria possess a structural weapon that makes them uniquely lethal.
The outer membrane.
Yes, that lipid-rich outer membrane that the alcohol dissolves. It is heavily studded with a molecule called lipopolysaccharide, or LPS.
And LPS is, fundamentally toxic to the human immune system.
Highly toxic. It acts as an endotoxin. But the crazy thing is, it isn't a poison the bacterium actively secretes to hurt you. It's just a structural component of their outer membrane.
But our immune system is just hypersensitive to it.
Exactly. If you have a severe gram-negative infection and you hit it with antibiotics, the drugs do their job. The bacteria pop.
But as they burst,
they shed massive amounts of that LPS structural debris directly into your bloodstream.
so the debris itself triggers the crisis.
Massive systemic inflammation. The immune system detects the free-floating LPS and just launches a catastrophic overreaction. It leads to a cytokine storm, blood pressure plummets, organs begin to fail.
Endotoxic shock.
Yes. You can successfully kill all the bacteria and still lose the patient entirely because of the architectural debris left behind by those thin-walled gram-negatives.
That is so intense. Okay, so we have the blueprint. We know the architecture of both types of walls. We know how antibiotics target them, and we know how our immune system reacts. But if the mechanisms are so well mapped out, Why are bacterial infections becoming harder to treat?
Well, because the cell wall is not a static blueprint. It is the active front line of a relentless evolutionary arms race. But before we even look at how bacteria mutate, we have to avoid a trap the sources call taxonomic overreach.
Right. Assuming that gram-positive and gram-negative represent this clean, perfect evolutionary divide in all microbes.
Exactly. It's a structural test, not a universal rule. Nature constantly breaks the binary. Look at mycoplasma, for example.
The bacteria responsible for walking pneumonia. Right?
That's the one. They evolved to ditch the wall entirely. Zero peptidoglycan.
Just a naked cell.
Yep. They survive by stealing cholesterol from their human host to stabilize their naked membranes. And because they lack the pressure vessel, penicillin is chemically useless against them. There is literally no wall to target.
Mind-blowing.
Mm.
And then you have Mycobacterium tuberculosis, which is structurally related to gram-positives, but it coats its peptidoglycan in this incredibly dense, waxy layer of mycolic
The
acid.
gram stain just bounces right off it.
Right. And you even see completely different domains of life arriving at similar solutions. Archaea, which are genetically distinct from bacteria, often build pseudo-peptidoglycan walls.
Right. They stain gram-positive, they form a pressure vessel, but the underlying chemistry is completely different. It's a textbook case of convergent evolution.
Nature finding the exact same structural solution from a completely different starting point. But, the real tension here isn't just these evolutionary outliers. It's how standard pathogenic bacteria actively rewrite their wall architecture to survive our drugs.
The adaptations are staggering.
The classic example in the sources is MRSA, right? Methicillin-resistant Staphylococcus
Yes.
aureus.
Yeah, MRSA is the perfect example. For decades, we just saturated the environment with beta-lactam antibiotics. The evolutionary pressure on these bugs was immense.
Adapt or die.
Exactly. So in response, strains like MRSA evolved an altered version of that cross-linking enzyme. They use a mutated transpeptidase called PBP2A.
Okay, so how does PBP2A help them survive?
It still builds the peptidoglycan wall perfectly, but the physical shape of the enzyme's active site is just slightly warped, so the antibiotic no longer fits the lock. It simply bounces off while the enzyme just keeps building the armor.
Wow. But I have to say, the most staggering example of this arms race, like the one that really got me into reading, involves VRE.
Oh, vancomycin-resistant enterococci.
Yes. Which emerged as a major hospital threat in the 1980s because vancomycin is a massive, complex molecule. It's our heavy-duty last resort antibiotic.
Right, and it works differently than penicillin. It doesn't bind to the enzyme. It actually binds directly to the wall itself.
Specifically, it caps the very end of the peptide chain on the unlinked wall precursor. Like the end of that chain naturally features two D-alanines stuck together, right?
Yeah.
D-ala-D-ala.
Yep, the D-ala-D-ala tail.
And vancomycin just snaps over that tail like a padlock.
Okay.
It physically blocks the cross-linking enzyme from getting close enough to stitch the wall together.
It puts a massive physical cap on the construction site. So how do the bacteria get around a physical blockade like that?
This is the crazy part. With the mutation of terrifying minimal precision, the resistant bacteria altered their internal assembly line to swap out exactly one atom in that tail.
This one atom?
They changed an amide bond to an ester bond, just to picture the mechanics of that.
Yeah.
An amide bond provides a crucial hydrogen atom that sticks out and acts like a magnetic grip.
And the vancomycin relies completely on that hydrogen atom to lock into place.
Exactly. So by swapping the amide for an ester, the bacteria replace that hydrogen atom with an oxygen atom.
The magnetic grip is instantly gone. They essentially replace the final D-alanine with D-lactate. So D-ala-D-ala becomes D-ala-D-lac.
And structurally, the overall shape is almost identical, right? So
Yeah.
the bacterial enzymes can still cross-link the chainmail just fine.
Yes. But because that single hydrogen atom is missing, there is literally nothing for the vancomycin to magnetically grab onto.
That one atomic tweak reduces the binding affinity of the drug by 1,000-fold. Vancomycin just slips right off the wall.
A single shifted atom defeats a last resort antibiotic. It really highlights how microscopic and mathematically precise this battlefield truly is.
That's terrifying, honestly.
The cell wall is just a dynamic high-pressure zone constantly being remodeled, and it is actively resisting our best chemical weapons.
Man, so just tracing the path we've taken here, we started with a frustrated doctor in an 1880s morgue using dyes to inadvertently expose a massive architectural divide in the microbial world.
A real happy accident.
Yeah, and then we zoomed in to see a continuous spherical chain link fence woven from mirror image wrong-handed amino acids.
The chainmail onesie.
The onesie. We watched penicillin cause molecular traffic jams that forced bacteria to pop from their own internal turgor pressure. And finally, we explored how swapping a single atom can shift the balance in a global evolutionary arms race, rendering our strongest drugs completely useless.
It's a lot to take in, but taking all of that and connecting it to the reality of the human body, there is a lingering thought here that fundamentally changes how you perceive your own immune system.
Oh, lay it on me.
Throughout this deep dive, we've focused on how breaking down the wall kills the bacteria.
Right.
But the sources point out that the human innate immune system has evolved specific sensors, they're called NOD-like receptors, that are perfectly tuned to detect the broken fragments of peptidoglycan.
Wait, so our bodies are looking for the wreckage?
Exactly. Think about the implications of that for your daily life. Every time you take a breath, or eat a meal, or just touch a surface, you are interacting with millions of bacteria.
Sure.
Even if those bacteria are entirely dead, even if they've already been shattered into microscopic pieces by the lysozyme in your saliva.
Your immune system is constantly reading and reacting to the broken fragments of that chain link fence.
Your cells are continuously detecting the microscopic ghosts of this armor.
The ghosts of the armor. Wow. So this high-pressure war we've been breaking down, it is happening inside you right now on a chemical level, whether you realize it or not.
Every single second of every day.
That is something to think about the next time you sneeze. Well, thank you so much for joining us on this deep dive into the hidden high-pressure world of the bacterial cell wall. We will catch you next time.